So 9.2 is focusing on the transport within phloem vessels, and phloem, like xylem, is very specialized in terms of its structure to be able to perform its function. The electron micrographs here, you can see that they are unusual shapes, and we'll break that down in just a moment. Now, the function of phloem is to transport carbohydrate around the plant. Specifically, it's transporting sugar in the form of sucrose. Now, sucrose is a disaccharide. It's made up of two individual glucose molecules which are bonded via a glycosidic bond. So connect all the ideas here to what we've done in the earlier units in Unit 2. So sucrose is very soluble, and it's dissolved in water, and it's moved through these phloem tubes, usually in the direction from the leaf, which is referred to as the source, because that's where photosynthesis takes place, and it's going to be moved to different parts of the plant referred to as the sinks, notably the roots, because the roots, of course, can't do photosynthesis, but they still need to have a supply of carbohydrate, of glucose, to be able to do respiration. Have a look and check through these syllabus statements and make sure that you go back to those at the end. Here, this is a bit of a repeat from what we did in 9.1. You can see the tissue plan of a dichotomous stem, and you've got these vascular bundles which are found in the dicot in the periphery here. On the outside, noticeably in pink, you've got the phloem tissue, which is what we're going to focus on in a moment. And then inside, we've got the xylem, and then we've got this cambium ring. Cambium, remember, is the meristematic tissue. That means it can undergo cell division, mitosis, and that increases the girth of the plant. If we look at the stem as well, we can see this inside cell layer, which is called the pith layer, is basically unspecialized, and we've got the cortex, which are more unspecialized cells, tends to be smaller in nature around the outside. Here we can see the actual cell micrograph and the staining, and what you need to notice here is that the size of the phloem is very different from the size of the individual xylem vessels. So the phloem is up here in this section of the diagram. Again, here we've got same sort of thing, but this time you can see that the vascular bundles are dispersed all over the place. So this is a monocot or monocotyledonous plant as opposed to a dicotyledonous plant. Now, I'd spend some time pausing and taking a really good look at this slide because it's probably unfamiliar. What we can see in blue is the sieve element cells, and the sieve element cells are what makes up phloem really, and it's inside these blue stained vessels here that we can have the transport of sucrose. Now, accompanying that vessel, we've got here in yellow a companion cell, and the companion cell, as it says, is the life support system for this blue sieve element cell. Now, the blue sieve element cell has got reduced cytoplasm, and it's also got very, very few organelles, so it hasn't got much of a metabolic capacity to do metabolism. By contrast, the companion cell does. So the companion cell is going to do most of the chemical reactions to support the life processes of the sieve element cells. But the sieve element cells are alive, which is different from the xylem vessels. What we've got in green is packing tissue called parenchyma. And parenchyma or parenchyma, depending on how you pronounce it, is very unspecialized packing tissue. The red here is sclerenchyma, and they are fibrous cells which are very, very important for the rigidity of the stem. So the more sclerenchyma fibers that you've got, the stiffer the stem will be. Remember with the parenchyma to link this to this idea of turgor pressure, so that the cytoplasm, which is full of water, will swell. It'll push the cell membrane against the cell wall. So the turgor pressure will give a lot of support to this part of the plant, but these sclerenchyma will add additional support. If you look at this diagram, what we can see now, again, is the sieve element cells here, and in cross-section, joining one to the next, we've got what's referred to as a sieve plate. And the sieve plate is cross-section here. You can see it's got tiny perforations. So what you've got are strands of cytoplasm which can actually run straight through from one cell into the next cell. So the sucrose is transported from one sieve element cell to the next cell through these sieve plates. And the reason it's moved is because of the development of a very, very high pressure from one end to the next end. So you're going to get the movement of sucrose from high pressure to a lower pressure. And we'll come back to this idea of that hydrostatic pressure generation in just a moment. But What's important on this slide is to appreciate that you've got these sieve plates at this surface here. Also, the cell walls around the outside, not the horizontal, but the longitudinal side, is impermeable to water. So that's very strengthened with cellulose within the cell walls just here. I would recommend that you dig into the Google Classroom and that you actually play this for yourselves. And this McGraw film is very, very good to see things that are moving. So I would encourage you to pause here, open up these documents, have a watch of those, and then come back to this document, which is essentially a summary. So hopefully the McGraw clip gives you that nice visual there, but what we've got to focus on is this idea of active translocation. So active means it requires ATP. So plant tissues at the source have a high metabolic rate. The companion cells are very much involved in the production of vast quantities of ATP, which is actively pumping the sucrose at the source into these phloem sieve elements. As water follows by osmosis, you get the generation of a high pressure, high hydrostatic pressure at the sink area, and that's going to force the movement. Now, what we're talking about the movement from source to sink. So the source is essentially the leaves of the plant, and the leaves of the plant are producing carbohydrate from photosynthesis. Also within the leaves, you've got the development of amino acids. So you've got, remember, nitrate being taken up at the root. It's being, again, dissolved in the xylem, taken up to the plant, and the plant can do protein synthesis. And again, link this idea of protein synthesis to everything that we've done in Unit 8. So we've got transcription and translation and all that good stuff. So the sink tends to be areas of the plant which require sucrose, which is broken down into glucose and then used for respiration. So growing, anything that requires energy is going to require ATP production, and the sinks are essentially the root of the plant. It could be lateral branching of stems, the development of fruits, and also things like potatoes, which are called tubers, or rhizomes, which are artificial roots. This diagram is also a further explanation of the same sort of thing. You've got the leaf here doing photosynthesis, the leaf making carbohydrates, it's making glucose and sucrose. It's actively pumping that into the sieve element, and water following by osmosis will create this big positive hydrostatic pressure here, which is going to move the sucrose in that direction. Same is true for amino acids as well. So we've got amino acids moving and water following by osmosis. This diagram again is showing you much the same thing. It's emphasizing that we need the companion cell here because the companion cell is responsible for the production of ATP, and the ATP is necessary for active transport. This diagram also is reminding you of the fluid mosaic model and this idea that we've got proteins which are serving all sorts of functions, including the production of ATP, ATPase, and also the actual pump itself, which is moving the sucrose by active transport. So again, that's unpacking much the same thing. Here, we've got the same sort of idea. We've got the high hydrostatic pressure being generated. It's pushing that and actively unloading the carbohydrate at this end at the sink. So you've got active loading and active unloading subsequently at the other end. This particular syllabus statement is linking the idea that you've got data that allows us to unpack and produce theories that is how we know about the movement of carbohydrate. So the aphid, which is shown in the diagram here, has got a structure which is called a stylet. And the stylet is a piercing instrument. And what this insect does is it punctures the plant tissue and it sucks the sucrose solution from inside the phloem tubes. So what we can do with this idea is carbon date and carbon label, radioactive carbon label of carbon dioxide that the plant is exposed to, and we can track the fact that the carbon dioxide is taken up here. Once it's been taken up, it'll be used in Calvin cycle. So again, link everything that we're talking about here to photosynthesis at a higher level. and we got the production of carbohydrate, which is then moved through the plant. And you can see that that trace is all the way eventually down into the root system. But by doing this and puncturing the plant at various positions using an artificial stylet and collecting that radioactivity, we can track the movement of carbon molecules through the plant. Could be linked to a data analysis question here. Yep, so this is just effectively saying that you've got radioactive labeling of compounds, carbon-14. So I think we've covered all of the syllabus statements. I will recommend that you go back, check in one by one, make sure that you've got this unpacked. The terminology and the connections to other parts of the course are the only thing that I see as being complicated, and then post me some questions on the Google Meet.